What are the basic steps in gene cloning? The basic steps in gene cloning are isolation of gene of interest. For this, we use restriction endonucleases. With the same restriction endonucleases, we have to cut the vectors. Here, the vector is a plasmid. And if we use the same restriction endonuclease, it will leave sticky ends in the plasmid and as well as the gene of interest. With the help of DNA ligase, we have to insert the gene of interest into the cut DNA sequence. Now, the formed DNA is the hybrid DNA. It is also called chimeric DNA. It has the plasmid DNA and as well as foreign DNA, transferring of recombinant DNA molecule into the host cell. This process is called transformation. Later, the selection of recombinant transformants. What are recombinant transformants? The host cells which received rDNA are called recombinant transformants, allowing the host cell to multiply to produce rDNA copies. At last, purification and extraction of the product. These are the basic steps in the gene cloning.